Yeah, so the journey of a catalyst is about the development of a customized chiral catalyst that enables our customer, Bayer Crop Science, a ton-scale production of an agrochemical. But before I go into this specific story, I want to give a brief introduction. Why we actually do metal catalysis and what matters there. Well, the direct answer is pretty simple. We want to make small organic molecules. And really the conceptual groundbreaking work was done about 130 years ago by Wilhelm Ostwald and others. And they figured really very nowadays simple concept that the catalyst is not used up during the reaction. It doesn't change actually the energy of reactants and products, the potential energy. What it does is lowering the activation barrier. And this concept then pretty quick made it actually to industrial processes. So in 1909, when Ostwald received his Nobel Prize for his groundbreaking work, Haber was already able to extract nitrogen out of the atmosphere and produce per hour 125 milliliters of ammonia. And this then quickly became the Haber-Bosch process. In just a few years, ton-scale production of BASF making ammonia. So a small molecule with a huge impact. It's a fertilizer and enables to actually feed the world. So it defines what we actually have to eat, and even us. So about 80% of nitrogen in our bodies went through that catalytic process and touched that catalyst. So this heterogeneous catalysis, as you can see, rather harsh conditions, high temperatures, high pressures. Still, heterogeneous catalysis is used also in fine chemical industry. But today I talk more about homogeneous catalysis. So milder, highly specific reactions that enable us to build complex molecules. So we can do cross-coupling reactions such as this Buchwald-Hartwig amination to attach certain residues to aromatic cores, or asymmetric hydrogenations, the topic of today, here an asymmetric enamide hydrogenation to make chiral enamines. So just one enantiomer that then is the active pharmaceutical ingredient. And temperatures are lower than 130 degrees. And the catalysts here actually, they are made of often precious metal, palladium, rhodium, or iridium, and a ligand. So the ligand solubilizes the metal, so it's in the same phase as the starting materials, and it tunes its reactivity by electronic and steric factors. And by that tuning, we can then make very specific reactions, transformations, and it's always really substrate dependent what catalyst works best and what conditions, solvent, additives, and so on are used. And at Solvias, we screen these different catalysts to then find a perfect solution that enable them to actually produce these molecules. So this is a selection of these complex molecules, and the tools that we have in hand also by catalysis actually shape what APIs are on the market and what molecules we can make, as well as the analytic methods nowadays allow us to understand what we actually have in the flask and what we actually make to then manufacture in the market. It's always a challenge then to manufacturing this then in big scale to reasonable price. So yeah, so metal prices and supply chain security is certainly a topic that arises when we think about these precious metals. So precious metals are actually produced as a mere co-product of earth-abundant metals that are mined in multi-ton scale and at a completely different price range. In the meantime, we can also do homogeneous catalysis using these metals, but most processes actually rely on the precious metals. And actually the driver of this price development is not the fine chemical industry, it's much more other industry, electronic industry, but also automotive industry. And you see here, for example, rhodium got more and more expensive since 2050. That's why since fume catalytic converters actually for diesel engines are used more and more and rely on rhodium. And then just 80% of the most expensive metals coming from South Africa. At the beginning of the COVID crisis, then one facility closed, and this led then to this price spike. So this is quite concerning. So it's good when we can then find solutions to lower the amount of the metal that we need actually for production. And at Solvias, we do this. We have a long-grown expertise to have a high starting point, and you will see later what that means. And we have a large catalyst library of commercially available catalysts, as well as of research ligands and catalysts that we can employ for a certain problem and to solve the problem of the customer. And we have a state-of-the-art automation. We just installed a robotic high throughput experimentation platform that has automated access to more than 1,000 different compounds around the clock. And solid dispensing makes it very flexible. And by that, we very fast can identify the ideal catalyst for the problem that the customer has. So we do the lead identification there, then adapt and optimize conditions to lower the catalyst loading, which is then key to make it feasible on scale. And the story today is even about after we identified a lead that was available, we customized that ligand more to make it more active, and then can even on ton scale for agrochemistry scale that process. Besides, we also use that platform to develop earth-abundant metal catalysis. Just as a side note, manganese catalyst, for example, we developed in collaboration with the University of St. Andrews, the Clark Lab, to replace stereometric reductants in ester hydrogenation and ketone hydrogenation. We can use, in the meantime, cheap cobalt-2 salts for combinatorial screening to do asymmetric hydrogenations and complement or replace rhodium and expensive iridium catalysts. And then there's also copper that complements in cross-coupling the established palladium catalysts. And again, key is really not so much if it's an earth-abundant metal or a precious metal. It's more to lower the catalyst loading to really low levels. And this I like to show you in a big case that was developed in the Siga labs. So these are now the Solvias laboratories, catalysis laboratories from the 80s until end of the 90s. And metolachlor as a herbicide that was used in the US already since the 70s as a racemate. However, beginning of the 80s, the scientists figured it's actually just one enantiomer that's the active ingredients and responsible for the positive active on the field. And then, well, the idea is develop an asymmetric hydrogenation. Don't make the racemate, just adjust the active compound. And by that, you can halve the amount of herbicide on the field, you halve the cost, and you have a huge positive economic and environmental impact. And this herbicide is still 2019, where the last data that I found used in multiple ton scale on the field. So big impact, just using half, have to produce half of it. There's an asymmetric imine hydrogenation. And for that, a whole new class of chiral ligands was developed, diphosphine, ferricene-based catalysts. And with that system, you just need 34 grams of iridium precursor and 68 grams of ligand to make 10 tons of this amine. So this is then feasible and still up to date, the largest asymmetric hydrogenation on scale, homogeneous catalysis. And this was actually the foundation of the Solvias ligand catalog. So product business, and more derivatives were added over the years and enable agrochemistry production, but also the production of APIs or functional materials. And in a collaboration with the Pfaltz lab in the 2000s, then a new class was added, the Uber-Fox type iridium complexes. And this was the point then when Bayer approached us also with that problem, with their problem. They want to do an asymmetric hydrogenation of an unfunctionalized, not directly conjugated alkene, highly hindered, and for that these catalysts are known to be the ideal solution. So it's not as easy as an enamide hydrogenation, for example. And they had this challenge. They actually wanted to bring a new product to the market, a novel fungicide. And also here, just one enantiomer is the active ingredient, not a racemate. And they had good access to the starting materials, used a heterogeneous catalyst first, palladium on charcoal to make a racemic compound, and then try to do a chiral resolution with recycle of the andesite enantiomer, also with heterogeneous catalysis, harsh conditions. However, this was not economical, and they couldn't realize that on ton scale. And here, asymmetric hydrogenation, homogeneous catalyst would deliver just the needed enantiomer, and they could skip with that the resolution, decrease energy consumption, decrease waste production, and enable then economical, scalable green process. And this idea was not new. Already other company, Sumitomo, did high throughput screenings actually, and they looked at ruthenium, at rhodium, and also actually at the Solvias iridium catalyst. So 3 are only on the market enabling this catalysis. And they got, well, the best results with this iridium Uberfox complexes, but not good enough, like limited conversion around 60% EE, enantiomeric access of 71%. But it was a start. They did a patent application, but was never realized on scale. And at this point, Bayer approached us, and we did one high throughput screening, 96 reaction on a well plate. So the scale is one of these reactions, 0.5 milliliters, so very small. And there we test different catalysts that we had available, also research catalysts, different solvents, additives from our experience. And then we generated leads. And this is what I mean with a high entry point. What we get out of here is then the first results. And we found actually that we can achieve with the acetylated starting material full conversion and high EE, so 98% enrichment in 3-fluoroethanol. So these screenings are done under ideal conditions and at high catalyst loading to not miss a hit. So the next task is then to lower the catalyst loading. So you can't work with like 2% more, for example. You really have to lower the loading. And this is then done by altering temperature, solvents, substrate concentration, and so on. And with this found hit here, that's commercially available from the catalog, we could reduce the catalyst loading to an S2C of 1000. That's 0.1 more percent. In hexafluoroisopropanol, so full conversion after reaction overnight. Not many side products. And at that point, well, that's pretty nice. For API production, that might be already good enough actually in catalysis. But Bayer said, well, our end product actually is $10 per kg or less than $10 per kg. So we can't realize that on scale. It's not, you can't afford it. You would use like the worldwide yearly production of iridium to realize that. And we try to go lower, and you see lower S2C, no full conversion anymore. And this was then the point where Bayer Crop Science approached an academic research lab, the Bella Lab at Likhat Rostock. And their PhD students did an alteration actually on the backbone of this iridium complex. So they altered the 2 position and the para position, and they found, well, auto substituents are of advantage, give a more active catalyst. In the 4 position, para position, methyl is better than nothing, or phenyl. And then on the phosphorus, instead of terzbutyl groups, the cyclohexyl group. And with that, they got a more active catalyst, S2C of 10,000. So 10 times less iridium needed, 10 times less ligand needed, 10 times less cost for that key step. What they found overnight, no complete conversion, 40 hours, still no complete conversion. So it's reasonable, but not good enough. And then additive screening was done. And at Solvias, this process as proof of concept was scaled to 1 kg in an 8-liter autoclave reactor. And with that, S2C of 10,000 plus an additive and Lewis acid, BF3 etherate, also in catalytic amounts. After 16 hours, full conversion and high enantioselectivity was achieved. So later it was actually found, even by looking at the kinetic data, that after 4 hours complete reduction was achieved, so even more active. So probably this will allow to even go lower in the future. And here you see the safe handling of HFIP. This can be handled in manufacturing factories well, but in laboratories you need to be really careful. And you see here 80 years of experience. Hans Meyer is now retired, worked more than 50 years in the Solvias labs, and Romano Grande, 30 years experience in that kind of chemistry. So they really realized this then in the lab. And with that, Bayer said, well, this is now a process, this we can commercialize at that point. But we used actually an iridium complex that just was done in the research lab in little gram amounts in the university. So you also have to make that, right? And this is where then also biochemists, process chemists, as well as the Solvias process chemists and the catalysis lab work together to produce actually that ligand, and that to make that on kg scale, that then enables to make the agrochemical on ton scale. And there were certain difficulties in that synthesis. Column chromatography, not possible on scale. Reactions, for example, at -78 degrees, a Suzuki cross coupling in the first step at high catalyst loading with an expensive catalyst. And this was also solved. Today, I just talk about this last step, an asymmetric reduction of a ketone that established the chirality in the backbone of the ligand, that's then transferred to make only the desired enantiomer. So also a key step. and there was no efficient method reported. They did this actually with quantitative reductants in the research labs. And in the area of ketone asymmetric hydrogenation, there's more done, and it was decided to do an asymmetric transfer hydrogenation. And available ruthenium complexes were scaled. Again, 96 reactions, different solvents, reducing agents. We got many leads here, and the 3 best were picked up, tried in the lab, and then reaction monitoring allowed to then find that TS-DANAP, commercially available ruthenium complex, is able to do this quite effectively, and further refining then allowed to get a higher EE in acetonitrile and a loading of 0.5 more %. In this case, we just need to make kgs of that compound, so that's reasonable to be then scaled. And also the other factors were then solved by Solvias process chemists and Bayer process chemists together. So at the end, 3 simple solid isolations and an overall yield of overall this step of 68% to make this backbone. Then the end game, phosphorylation. The ligand group at Solvias is very experienced and set up to handle phosphines and phosphine chlorides, also on large scale, and reagents such as N-Buli. So they did the phosphorylation, attached directly, did a complexation in one pot with iridium cod chloride, sodium bath to give them this beautiful crystalline bright red catalyst that you actually can, so that's then air stable in contrast to the phosphines that are highly reactive. So air stable compound that can be well employed at a factory. And with that, Bayer now produces this intermediate for their fungicide on ton scale. Further optimization in the loop reactor then allowed in the meantime to lower the catalyst loading even more, and Solvias provides this catalyst enabling this chemistry. Yeah, here you see sustainability gains, PMI index -70%, energy consumption was reduced by -26%, and CO2 equivalence by 45%. So sustainable and economical process, bringing then this product to the market. And the development of this technology then can also be translated to pharma products and API production. With that, I come to an end. I really thank the whole team Bayer Crop Science, and in particular Christoph Schotes, who was the driver behind that project, the team Likard Rostock, the research lab of Matthias Beller, and then the team at Solvias that enabled to realize that process in collaboration with our customer and the university group. And thank you very much for your attention, and I'm happy to answer your question.